Kia ora team, welcome back to the 2.5 Genetic Variation and Change 4-credit external video series. This is video 10. In this video, you'll be learning about dihybrid crosses. And by the end of this video, you should be able to identify the expected phenotypic and genotypic ratios of dihybrid crosses using Punnett squares, and relate the inheritance patterns seen in a dihybrid cross to the Law of Segregation and the Law of Independent Assortment. So let's recap. Previously, you've learned that genes are sections of DNA that code for proteins and that they come in different versions called alleles. Individuals inherit one allele from each parent for each gene. You've also learned that the Law of Segregation states that two alleles for a gene segregate or separate during gamete formation, and they end up in different gametes. This means that at the end of meiosis, each gamete only gets one allele. You've also learned that the Law of Independent Assortment states that the alleles of a gene segregate independently of all other alleles from other genes, if those genes are on non-homologous chromosomes. You've also learned that monohybrid crosses are used to study just one gene with two alleles, for example, pea plants with tall versus short stems. And you've learned how to draw and use Punnett squares to predict the genotype and phenotype ratio of resulting offspring from a monohybrid cross. Finally, a monohybrid cross Punnett square is 2 columns by 2 rows, because each parent produces 2 possible gametes. 2 times 2 is 4, hence there are 4 squares in the middle to show the 4 possible offspring. So what's a dihybrid cross? A dihybrid cross is a mating between 2 individuals where the inheritance pattern of 2 genes are studied. For example, let's say the first gene is gene A, and the second gene we're studying is gene B. Remember that each parent contributes one allele for each gene to its offspring. So this means that each parent can produce 4 different types of gametes, not just 2. These 4 row headings show the possible allele combinations that parent 1 can produce in their gametes after segregation has happened at the end of meiosis. And the 4 column headings show the 4 possible allele combinations that parent 2 can produce in their gametes after segregation has happened at the end of their meiosis. Because of this, the Punnett square for a dihybrid cross is 4 columns by 4 rows. 4 times 4 is 16, hence there are 16 squares in the middle to show the possible offspring. You then have to fill in the squares like you normally would for a monohybrid cross. It's just this time around you're dealing with 2 genes, gene A and gene B, instead of just 1 gene. And 4 alleles for each zygote, big A and little a, and big B and little b. Whereas in the monohybrid cross, you only had to deal with 2 alleles, because you were only looking at 1 gene. If this is all getting confusing, just please hang in there until the end of the video because I'm going to go over the steps more slowly and in more detail. After that, you then have to count the genotypes to be able to write the genotypic ratio. And then you have to identify the phenotypes, count them to be able to write the phenotypic ratio. So there are 7 steps you have to follow. Let's go over them step by step over the next slides. Okay, here is the information that will help us for our guided practice today. In pea plants, one gene controls flower position while another gene controls the flower color. The 2 genes are unlinked. Here's a table showing information on the trait, the dominant allele, and the recessive allele. Mrs. Heald, that's me, crossed 2 pea plants that were heterozygous for both traits. State the expected phenotypic ratio of the offspring produced by this cross. So the first step in doing our dihybrid cross is to identify the 2 genes and their alleles. Decide which allele is dominant and which allele is recessive. The information tells us that 1 gene controls flower position. So that's our first gene, flower position. Our second gene is the gene that controls flower color. so we put that information up here. Now let's have a look at our alleles. For flower position, our dominant allele is axial, capital A, and our recessive allele is terminal, little a. For the gene of flower color, our dominant allele is purple, big B, and our recessive allele is white, little b. It's important to extract specific information from the question you're given and put it into your own grid. Step 2 is to write the genotypes of the two parents. Our information tells us that both parents were heterozygous for both traits. So that means that their genotype is big A little a and big B little b, and big A little a and big B little b. So our two parents have the same genotype, and for both genes they have the dominant allele and the recessive allele as well. Step 3 is to determine the gametes each parent can produce. For this step, we're going to use the FOIL, FOIL method. Yes, like the method you use in algebra. Now, the information clearly tells us that the two genes are unlinked. This means that genes A and B are on different chromosomes. And so the big A and little a alleles are free to segregate into the gametes independently of the big B and the little b alleles. In other words, unlinked means alleles aren't linked or they're not tied up to each other because the genes are on different chromosomes. F stands for first. The first allele of each gene goes into the first gamete. That's the big A allele for gene A and the big B allele for gene B. So let's pop those into the first box for the first gamete. O stands for outer. The outer allele of each gene goes into the second gamete. That's the big A allele for gene A and the little b allele for gene B. Let's pop those into the second box for the second gamete. I stands for inner. The inner allele of each gene goes into the third gamete. So that's the little a allele for gene A and the big B allele for gene B. Let's pop those into the third box. And finally, L stands for last. The last allele of each gene goes into the fourth gamete. So that's the little a allele for gene A and the little b allele for gene B. Step 4. Set up a 4x4 Punnett square. List one parent's gametes along the top and the other parent's gametes along the side. Remember, these gametes have just come from these boxes that we just did for step 3. Step 5. Fill in the boxes by combining the alleles from each gamete in the row with each gamete in the column. So these alleles from this row combine with these alleles from that column. These alleles from that row combine with these alleles from this column. These alleles from that row combine with these alleles from this column. And these alleles from that row combine with these alleles from the same column. I think you get the point, so I'm not going to continue with the animations. Here are the 16 completed boxes, with each box representing the genotype of a possible zygote that could be created after a fertilization event. Notice how the genes always stay in alphabetical order, A over B, A before B, and if there is a dominant allele, the dominant allele is always to the left of the recessive allele for that gene. But the genes always stay in alphabetical order. And sticking to the system keeps your Punnett square organized and reduces the likelihood of you making mistakes. I also want to point out that each box represents the male gamete fertilizing the female gamete. The gametes are haploid because they only contain one allele for each gene, so one set of chromosomes. And when the male and female gametes combine during fertilization, they produce a diploid zygote. diploid because there are now 2 alleles for each gene, 2 sets of chromosomes. Step 6. Count the genotypes so that you can write the genotypic ratio. I've used colors here to make all of the genotypes easier to see, and therefore easier to count, and therefore easier to write. And finally, the last step, step 7. Identify the phenotypes, count them so that you can write the phenotypic ratio. I've used colors here too to make all of the phenotypes easier to see, and therefore easier to count, and therefore easier to write. So going back to our original question, state the expected phenotypic ratio of the offspring produced by this cross. And here is our final answer. This cross produces 9 plants out of 16 with axial flower positions and purple flower color. 3 plants with axial flower position and white flower color. 3 plants out of 16 with terminal flower position and purple flower color. And finally, 1 out of 16 with terminal flower positions and white flower color. I want to draw your attention to this phenotypic ratio. When 2 individuals that are heterozygotes for both genes are crossed, and those 2 genes are unlinked, the phenotypic ratio will always be 9:3:3:1. This 9:3:3:1 ratio means that 9 out of the 16 offspring will display the dominant allele for both traits 1 and trait 2. 3 of the 16 offspring will display the dominant allele for trait 1 and the recessive allele for trait 2. 3 out of the 16 offspring will display the recessive allele for trait 1 and the dominant allele for trait 2. And finally, that 1 out of the 16 offspring will display the recessive allele for both traits 1 and 2. So, if 2 individuals that are heterozygotes for both alleles are crossed and the 2 genes are unlinked, the vast majority of their offspring should have the dominant phenotype for both traits, and only a very small proportion of their offspring should have the recessive phenotype for both traits. For example, in this cross, the vast majority of pea plant offspring should have an axial flower position and purple flower colors, while only a very small proportion of their offspring should have terminal flower positions and white flower colors. So in summary, there are 7 steps to completing your dihybrid cross. Step 1, identify the 2 genes and their alleles, which alleles are dominant, which alleles are recessive, and all of this information is given to you in the question. Step 2 is to write out the genotypes of the parents. Step 3 is to figure out the gametes each parent can produce using the FOIL method. F for first, O for outer, I for inner, and L for last. Step 4 is to draw up your 4x4 Punnett square and make sure you put the gametes along the sides and along the top. Step 5 is to fill in those boxes by combining alleles from the rows with the alleles from the columns. Step 6 is to count the genotypes and write that genotypic ratio. Step 7 is to identify and count those phenotypes so you can write your phenotypic ratio. So by now, you should be able to identify the expected phenotypic and genotypic ratios of dihybrid crosses using Punnett squares, and you should be able to relate the inheritance patterns seen in a dihybrid cross to the law of segregation and the law of independent assortment. Thanks for watching, and I'll see you in the next video.